do from the biomolecules, there is enzyme, okay, but this is enzyme. Okay, first of all, we will learn a little bit about the enzymes, okay, and then we will solve some question based on that. Okay. Enzymes, these are mainly protein, okay, these are proteins, but this protein has catalytic property. Proteins having catalytic property. Catalytic property means they increase the rate of reaction. They increase or they enhance the rate of chemical reaction. Okay, this and as you know, in chemistry also, I think you have learned about the catalytic agent, those catalyst. I think you know, no? Catalyst you have read in chemistry. Okay, those in chemistry that what we learn, those are the inorganic catalyst, and these catalysts are slightly different from the enzyme which function as a catalyst. Okay, here of course these are protein, so naturally proteins are with high molecular weight. This catalyst, that inorganic catalyst which is present in that is used in the laboratories and various chemical reactions, those are inorganic compound, and they work at a very high temperature again, very high temperature. But this, you know, our that enzyme catalyst, they have to work within a limited temperature, because our body temperature cannot rise beyond a particular temperature. This remain 35, 40. Okay, so within that temperature range that the enzyme have to work. All right. So these are proteins having catalytic, and as the inorganic catalyst also, these enzymes also, after carrying out the chemical reaction, the chemical reaction, they are unchanged. All right. And you know that within our body, in the biological system, there is a lot of continuous uh, uh, continuous process of the different chemical reaction, and all these chemical reaction, okay, they are catalyzed by enzyme. So enzymatically catalyzed chemical reaction continuously taking place within our body, and all those chemical reaction together we call what? We call the metabolism. Is it? Metabolism, all the chemical reaction. This how what happen with this enzymes, that the enzyme actually actually reduce the, that is, activation energy. Activation energy is the energy required for initiating a chemical reaction. This is energy required for initiating chemical reaction. So that for any chemical reaction, it is particularly if it is endothermic reactions or some, whether it is exothermics also, but to initiate first, it requires some energy, and that energy requirement is reduced. So as it reduces the requirement of energy, so the chemical reaction starts. Okay, so that enzyme carry out work by reducing the activation energy. All right. So the chemical reactions takes place, and what happen? The enzymes on which it work, the chemical compound on which enzyme work, that is known as substrate. Okay, this is known as substrate. So this substrate is, by this catalysis enzyme, this substrate is changed into product. All right, substrate is changed into product, and here is, but what happened then? The substrate combined with, if substrate I write with S, then substrate will combine with the enzyme, and then they form a transition stage. They form a transition stage, and that is a substrate enzyme complex. This substrate enzyme complex is a, this is a short period. This thing is for a short period, temporary, transient state, and then this one is changed into, this substrate is changed into product, then it will be product enzyme, uh, product enzyme complex, and then this product enzyme complex will separate, and it will be changed into product and enzyme. Enzyme will come out, but this one, this can move in both the direction. So naturally, to progress a chemical reaction, substrate enzyme combine with the substrate, form a temporary combined stage, and then substrate is changed into product. This is also for some time, it is very short period, and after that the product is released. So these things happen with the reaction of this. For these enzymes, no, there are many different types of chemicals, substrates are there, and so the number of enzymes also are lot. Many enzymes are there, and the enzymes are very, very specific. Enzymes are very specific. Specific in terms of the type of reaction it takes place, and then the substrate specific also, and the type of substrate, that substrate is specific also. Okay, so what happened for means to change the substrate into product, this enzyme has to catch the substrate. So there is in the all the enzymes, no, I have told they are protein, and protein usually they are functional in their tertiary tertiary structure. In the tertiary structure, that it forms a 3D structure, is it? And so the primary structure it fold in a different way, and ultimately secondary and then tertiary, and if it is required, then quaternary structure is also there. So when this tertiary 3D structure, tertiary structure is achieved, in that what happened, there are within the protein, there appear some crevices. Okay, some crevices, or we can say some pockets like a structure where this enzyme is fitted. Okay, when this enzyme is fitted to the crevices, then this uh, sorry, not the enzyme is fitted, that is that substrate is fitted to the enzyme pocket. Substrate is fitted to enzyme pocket. All right. Then this substrate is changed to the product. After that, substrate is changed to product. So this a special type of that crevices or pocket which appear in the 3D structure or the tertiary structure of the enzyme protein is called the active site. And these active sites are formed by some specific amino acids present in the protein. Okay, so it is active site. Active site is important. If there is change in the active site, then the enzyme will be functionless. Okay, as I told that the enzymes are protein, but what happened, the enzyme, most of the enzyme, only a few enzymes are there which are purely protein, but some most of the enzymes they have a non-protein portion. The enzyme enzyme have a protein part, protein plus a non-protein portion. This protein portion, this is actually main part, that is known as apoenzyme, and the non-protein portion is known as cofactor. Okay, so those enzymes which have both this apoenzyme and cofactor, okay, without this cofactor, the enzyme apoenzyme will not work. So this apoenzyme plus this cofactor, then it makes the holoenzyme, the whole complete enzymes. Then this cofactor again are of several type. Cofactor mainly that three type. One is known as prosthetic group. Prosthetic group. Another one is called coenzyme, and then there is metallic activator or the metal activator. Okay, this is prosthetic groups. They are organic in nature and tightly bound to the apoenzyme. Bound to apoenzyme. Okay, this is prosthetic group. So they are always remain attached to the main protein part. The coenzymes, they are also organic compound, but they are loosely attached. Attached, and it is transient. When the enzyme has to work on a substrate, the portion will come and join the apoenzyme, carry out the catalysis, and then again it will separate. Okay, that is apoenzyme. This organic this cofactor will come. Cofactor will come. You have heard about that NAD, NADP, DP, FAD. No, these are the cofactors. They are temporary. They have a temporary attachment with these apoenzymes. Then it makes the holoenzymes and it will work on this substrate. They will combine, then they will work on substrate and change the substrate into product, and after that product it forms this. From the enzyme the cofactor also will come out. Cofactor will not remain attached with the apoenzyme portion. So this is the coenzyme and prosthetic group difference is there. Uh, One of the very good example of the prosthetic group is the heme group. Heme is the prosthetic group in some of the enzymes for peroxidase, catalase, those enzymes. And then there is metallic activator. Metallic activator like sometime metallic activator like zinc. Okay, in some cases Mn, in some cases the magnesium. These metals, they function as the metallic activators. So the these are the metal activators. So this is enzymes, and enzyme has apoenzyme and cofactor, three different types. In the when you will go through that glycolysis, in that you will find those some of the enzymes they require the magnesium. In case of photolysis of water, manganese is required, and zinc is required for the carboxypeptidase. So these are the some of the. Then there is, as I told, this next is one thing is classification of the enzyme. Okay, in classification, but when I told that the enzymes are very specific for a particular substrate or specific for a particular type of reaction, right? So based on that, all the enzymes, according to, of course, convenient and the old system, the enzymes has been classified into several groups. Okay, these several groups they are the particularly this is oxidoreductase. One is oxidoreductase. As the term it has been, uh, you can see that it is oxidoreductase means the enzyme, this group of enzyme which carry out oxidation and reduction. Oxidation and reduction type of reaction it carry out. Okay, this one is also, it is that known as dehydrogenase. Dehydrogenase is also belongs to this group. Dehydrogenase enzyme, in case of wherever those NAD, NADP, NADH is transferred, in that cases enzyme dehydrogenase is used. Say in case of photosynthesis, there is a reduction of the phosphoglyceric acid into phosphoglyceraldehyde. There the triose phosphate dehydrogenase enzymes are required. So these are that oxidation reduction reaction it carry out. Next is, that is the transferase. This one is that catalyzing a group transfer. Group transfer between substrate from a particular functional group from one compound, it is transferred to the another compound. So this is called the transferase. After that, there is one very big group of enzymes are there, that is known as hydrolases. Hydrolase, this is hydro, water, and lases, okay, hydrolysis, as you know, that breakdown, lysing, that is hydrolysis. This hydrolases, they break some bonds, okay, dissociation of bonds with the release of water. This is this enzyme catalyze the hydrolysis hydrolysis of bonds. Bonds like ester bond, then like the peptide bond, glycosidic bond, you know, but that when those polysaccharide or the polymer, most of the cases polymers change into their monomers. During this, a molecule of water is released. Say polymer, this polysaccharide is there, that polysaccharide change into monosaccharides, or one molecule of monomer has to be removed. Then in that case, in polysaccharides, the bond is glycosidic bond is there. So if one sugar molecule has to remove from there, then the glycosidic bond break. Okay, or the formation of bonds takes place, then that during the formation of glycosidic bond, okay, yes, formation of the glycosidic bond, during formation water is released, and during breakdown water is added. Clear? During bond formation water is released, and during the breakdown of the bond the water is added. So this is hydrolysis, by the hydrolysis of ester, and that it can combine with those bond formation between carbon carbon, bond formation between carbon and halide, those chloride bromide, no, that halide, this, during this is there is a hydrolysis. Another, that next group is also similar, that one is lysases. Lysases, here also there is a breakdown, okay, Enzyme that catalyze the removal of group, but not by hydrolysis. That is group other than hydrolysis. This is, and during this, the double bond is produced. Then number 5 is isomerases. In isomerase, you know the isomers. So this is the during the formation of uh, this catalyze the isomerization reaction. It catalyze isomerization reaction. Like if glucose is changed to fructose, glucose 1,6-phosphate changed to fructose 1,6-phosphate. Glucose and fructose are isomers. So during that, isomerase enzymes are required. And then the last one, this is ligases. Ligation means sealing, attachment. So this one is the bond formation, catalyze linking. This one catalyze linking, linking together two compound, linking together two compound. You will say that in case of glycolysis, no, I'll give you one example. In glycolysis, that mm, fructose 1,6 biphosphate or bisphosphate, it change into dihydroxyacetone, dihydroxyacetone phosphate and phosphoglyceraldehyde. Ah, PGAL, phosphoglyceraldehyde. Okay, so that is this one is enzyme is of course this is double this one is a reversible reaction. This one is catalyzed by enzyme which is known as aldolase. Aldolase. This aldolase is help in forming bond between this phosphoglyceraldehyde and dihydroxyacetone phosphate. During glycolysis, the reaction moves to this direction. During the dark reaction, that is the during the dark reaction or the that is Calvin cycle, this one goes in the opposite direction. That there it is the synthesis. This aldolase is a ligase enzyme. Two molecules, this one and this one, are combined together to form fructose 1,6 biphosphate. So there is this type of enzymes are there. This ligases enzyme you will find during the formation of the bond between the nucleotides that there is in when there is a synthesis of DNA. During DNA replication also you will come across the ligases. So this type of things are there. As a one thing, all the enzymes, whatever is there, at the end of their name, that is there is a suffix ase. Okay, so ligase, uh, isomerase, ASE, isomerase, ligase, uh, lysase, ASE, the hydrolase ASE is there. So enzyme names transferase ASE. Enzyme names are with the suffix ASE. All right. There is the modern detailed classification of there, in which there is a four steps classification is there, that every enzymes their type of reaction they have given a number. So there is four numbers are there. Enzymes are given name one, one, two, three, one in each groups. That detailed classification we don't require now. Okay, so let us now complete some of the reaction. Okay, one some important thing is that the enzymes are very specific, and they work in a narrow range, in a narrow range or within a limit of this pH, temperature, etc. This is highly influenced by the temperature and pH. If there is a change in the pH, if there is change in temperature, there will be change in the enzyme reaction. So this type of things are very important. Okay, let us come to now we will solve some question. So here those one question, that is the question number, say this one read as question number 1. Those acids which are nucleic acids which behave like enzyme are known as, say enzymes are protein. This is uniformly, most of the enzymes are protein, but some nucleic acid, particularly the RNA, uh, in some cases they function as the enzyme, and in that case it is known as ribozyme. This is, you can take this one as an exception that all proteins, all enzymes are protein. The RNA as an enzyme, this is an exception you can take. Okay. Next one. Next question is the crevices of the enzyme in which substrate binds. What is it called? The second question, the crevices of the enzyme in which substrate bind. Just now I have told
Active site.
this is called the active site. Okay. So this if the active site and the substrate does not fit properly, then the enzyme will not function. Okay. Next question is Next question is the rate of reaction doubles or decreases by half for every degree change in either direction. Say I told that the enzymes are very sensitive to temperature. Enzymes are very sensitive to temperature. So temperature influence. So enzyme activity means its capacity to work, its rate of reaction. That one is change. Either it will, the rate of reaction will become double or it will decrease by half. In both the direction, rate increasing or rate decreasing by half for every dash degree change. Which one? Every degree centigrade change in either direction. That is, the rate of reaction will increase or decrease. Which one? Here 10 degree, 15 degree, 20 degree, 27 degree.
Degree.
Huh? Yes? Which one?
Then degree.
Which
Then
one?
is
Oh, I cannot
number
hear.
one.
Number 4. No, it is number 1. Okay? 10 degree. For every 10 degree increase in the 10 degree Celsius increase, the rate of reaction will increase by double or it will decrease by double. Say with the rise in temperature, say if the temperature is, say it is something is there in 5 degrees Celsius, the enzyme is working. And if you increase the temperature, the rate of reaction also gradually will increase. So rate of reaction increase with the rise of temperature. But this rise is not infinite. Indefinite rise will not takes place. Okay. A time will come, it will reach its maximum. Okay. The rate of reaction will be maximum, but after that, if you rise the temperature, then this rate of reaction will gradually decrease. This is due to that at high temperature, that the degenerate protein degenerate, and the enzymes are protein. So with the high temperature, their 3D structure, the tertiary structure will, third level structure will not remain. So, and then their function will decrease. And that is why, when we go to very high temperature areas, say in our country during the summer, if we are going to, say, to Delhi, uh, or to the Rajasthan there, and if there is a little bit dehydration, etc., also, sometime that hot wave, due to hot wave, as a loo, that so people die, is it? People die. Why? That their enzyme system does not work anymore. Beyond 40 degree, then the enzyme system will not work. So when the temperature goes very high, then it is risky, and that's why to maintain the body temperature, we have to take the lot of liquid, plenty of water. And when this water evaporate from the body, that is the sweating, and we pass urine, that the body temperatures, no, come out from the body, making our body cool. Okay. By taking lot of water, maybe we have to repeatedly, frequently, we have to go to toilet to pass urine, but that passing urine bring our body temperature down. So maintain it. We have to keep on taking the liquid and evaporate it as a sweat, or to pass urine and cooling our body. So these things happen, that for every 10 degree, usually it is considered. This is one of the question. Okay, here is the next question. Which of the following describe the given graph correctly? Say, here it is given a graph. This shows the reaction of the that is a change of into substrate substrate into product. Okay, but with that addition, whether enzyme is there or not. Say this one at this, this is y-axis, and this one is x-axis. This side is the reaction, the rate of reaction. Okay, and this side is potential energy. So this one is showing that two graph you can see, one is going very high, all right, and another one is little low. This one here is the substrate, all right. This things is showing that the how much energy is required. This side is it energy, potential energy to take the reaction rate of reaction to increase, the potential energy is to increase very high. This one, this one is what is called that the activation energy, right? So here in this one, the activation energy requirement is more, but here the activation energy requirement is less. This one is enzymatic reaction. Enzymatic reaction, and this one is without enzyme. Okay, so this substrate after this reaction it change into product. Here the level of energy you can see, energy level is up to here, but when it change into product the level of energy is very down. So this energy will come out as will be released, and that is the when in a particular reaction the energy is released, we call the exothermic reaction. So this one is explaining what. Number one, endothermic reaction with energy, endothermic reaction with energy. A in the presence of enzyme and B in the absence of enzyme. This is true that A is in presence of enzyme. That is true. B is in absence of enzyme. But this one is not endothermic reaction, because afterward when this product is produced, then the potential energy level is coming very low. It means it will release energy. So this one is a exothermic reaction. So here is exothermic reaction with energy, and A in the presence of enzyme, B in the absence of enzyme. So this one will be the correct answer. Endothermic reaction, it is not endothermic reaction. Exothermic reaction, A in the absence of enzyme, no. They see the difference that this much energy is not required when enzyme is added. So here this graph is from your book. Okay, this is the graph, the concept of activation energy. This is the potential energy. This is substrate S, and this is the progress of the reaction. Progress of the reaction, and here is the product. The here this one is activation energy with enzyme. This energy level requirement, no, is very less, but this one is activation energy without enzyme. So without enzyme this require lot of energy. Okay, So this is what we call the activation energy, the amount of energy required for the for initiating or for carrying out a chemical or a biochemical reaction. Here is a small another question. One question. Choose the correct option. I have shown this one. Enzyme plus substrate, temporary enzyme substrate complex, enzyme plus product, then enzyme product. This one, the correct one will be, say, correct one is this, enzyme and substrate. This forms a enzyme substrate complex that is transitory, and this one is in both the direction. It moves, then it forms the enzyme substrate complex. Okay, and after that enzyme and substrate is separated. So this will be the right choice. This is what during the enzyme catalyzed reaction, enzyme catalyzed reaction, these things are required. Okay, one more, the question from the diagram here is, they select the correct graph which shows the effect of temperature on the velocity of a typical enzymatic reaction. Say this is the velocity, this side is temperature, rise in temperature. Ah. So which one will be the correct? Temperature on the velocity, effect of temperature. Say, as I told, the enzymes are very sensitive to temperature. So with the rise of temperature, this one won't be. With the rise of temperature, the rate of reaction increases. But if it will continue, then the protein, that enzyme protein start degradation, and so the rate of reaction again it will fall. So this one will be the correct graph to show the um, effect of temperature. Similar way, this is the pH. pH also, this protein, I think in the structure of protein we have learned that protein structure, this change, protein function and structure change with the change in the pH. Function of the protein changes, amino acid. Okay, so that enzyme function at a narrow range of pH. So at the pH pH, the with the rise in pH, the rate of reaction will rise. But if you continue, then the rate of reaction will fall down. So there is a, this is the optimum pH. Okay. So this one, C is the answer. But next one is the enzymatic activity, relationship between the rate of enzymatic activity and the substrate concentration. What happens when the enough amount of, if enough amount of enzyme is there, okay, If enough amount of enzyme is there and you are adding the substrate to it, with less substrate, rate of reaction will be lower. If you add more substrate, the rate will increase because enough enzyme is there. So, and if you keep on adding, the rate of reaction will increase. The rate of reaction will increase. But after that, when all the enzyme molecules are occupied by the substrate, then by adding substrate also, the rate of reaction will not increase. So this is, see, one term is there, B max, where the velocity of the reaction is maximum. All right. And this one, Km, is called Michaelis constant. Okay, Michaelis constant at the half rate of reaction is there. So this one will be the answer. It will increase and gradually it will come to a flat. Of course, so this one also, but this one explain better. This one explain better. Okay, today it is time is over, so we will stop today, and in the next class we will continue. Okay, thank you very much.
Thank you, sir.